This is the continuation of endocrine system. Now we will see pituitary gland. The pituitary gland is a small pea-sized lump of tissue connected to the inferior portion of the hypothalamus of the brain. Despite its small size, the pituitary gland is actually two distinct glands. They are the anterior pituitary or adenohypophysis and the posterior pituitary or neurohypophysis. These two glands are made of different tissue, excited by different types of stimuli and secrete different hormones. There are many blood vessels surround the pituitary gland to carry the hormones it releases throughout the body. The pituitary gland is located at the base of the brain, attached to the hypothalamus superiorly by a stalk called the infundibulum. The pituitary gland is about 1 centimeter in diameter. It is situated in a small depression in the sphenoid bone called the sella turcica. Arterial blood is delivered to the pituitary gland via hypophyseal branches of the internal carotid arteries. Veins leaving the gland drain into the dural sinuses. The anterior pituitary and posterior pituitary lobe have different functions. The anterior pituitary gland is also called the adenohypophysis and is composed of glandular tissue. It manufactures and releases a variety of hormones. The function of the anterior pituitary gland is controlled by the releasing and inhibiting hormones of the hypothalamus. The anterior lobe has three regions. Pars distalis, these are the largest, most anterior part of the pituitary gland. Pars tuberalis, these are the extension that wraps around the adjacent area of the infundibulum. Next is pars intermedia. These are the slender and narrow band that borders the posterior lobe of the pituitary gland. This section may secrete two types of melanocyte stimulating hormone, also known as melanotropin. It stimulates the melanocytes of the skin to increase production of melanin, and the release of melanocyte stimulating hormone is inhibited by dopamine. Releasing hormones from the hypothalamus control the anterior pituitary's secretion and travel in the hypothalamus capillary network. These fenestrated capillaries contain structures that resemble spores and form the hypophyseal portal veins passing along the pituitary stalk to the capillary network of the anterior pituitary. The hypothalamus releases substances that are carried directly to the anterior pituitary via the blood. Releasing actions of the anterior pituitary are mostly stimulatory, although some have inhibitory effects. The primary and secondary capillary plexus, along with the hypophyseal portal veins, are the structures that comprise the hypophyseal portal system. Via this system, releasing and inhibiting hormones from the ventral hypothalamus circulate to the anterior pituitary and regulate its hormone secretion. Releasing hormones stimulates synthesis and secretion of one or several hormones at the anterior lobe, while inhibiting hormones prevents this from occurring. Numerous hormones of anterior pituitary, which are all proteins, regulate the activity of other endocrine glands. The first one is thyroid stimulating hormone. As its name suggests, it is a tropic hormone responsible for the stimulation of the thyroid gland. Thyroid stimulating hormone controls the secretion of thyroxine and plays a key role in the regulation of metabolic rate. Next is adrenocorticotropic hormone. It stimulates the adrenocortex, that is the outer part of the adrenal gland, to produce its hormone and plays a key role in regulation of the stress response. Next is follicle stimulating hormone. It controls the production of eggs in ovaries and sperm in the testes. Next is luteinizing hormone. It stimulates the gonads to produce the sex hormones, estrogen in females and testosterone in males. Next is growth hormone. Human growth hormone affects many target cells throughout the body by stimulating their growth, repair, and reproduction. It stimulates the growth of bones and muscles. It also stimulates the liver to produce somatomedins, that is a protein that promotes cell growth and division in response to stimulation by growth hormone. Next one is prolactin. It has many effects on the body, the chief of which is that it stimulates the mammary glands of the breast to produce milk. It is also responsible for changes in the maternal brain that are thought to help prepare for the demands of becoming a new parent. Dopamine is a primary neuroendocrine inhibitor of the secretion of prolactin from the anterior pituitary gland. Now we will see posterior pituitary gland. The posterior pituitary gland is actually not glandular tissue at all, but it is nerve tissue instead. The posterior pituitary is a small extension of the hypothalamus through which the axons of some of the neurosecretory cells of the hypothalamus extend. These neurosecretory cells create two hormones in the hypothalamus that are stored and released by the posterior pituitary. The connection between the hypothalamus and the posterior pituitary is very different from that of the hypothalamus and the anterior pituitary gland. Groups of neuron cell bodies in the hypothalamus produce antidiuretic hormone and oxytocin. These two hormones are delivered by axonal transport through the infundibulum to the posterior pituitary, where they are stored. Nerve signals from the hypothalamus travel through the infundibulum and trigger the release of antidiuretic hormone and oxytocin from the posterior pituitary gland when they are needed. Although these two hormones are technically made in the hypothalamus, they are referred to as posterior pituitary hormones because they are released from posterior pituitary gland. Antidiuretic hormone prevents water loss in the body by increasing the reuptake of water in the kidneys and reducing blood flow to sweat glands. Oxytocin triggers uterine contractions during childbirth and the release of milk during breastfeeding. How the pituitary gland secretion is controlled. The pituitary gland does not release a steady flow of hormones. Rather, it releases hormones in phases or pulses. For example, growth hormone is mainly secreted at night, whereas luteinizing hormone surges in the middle of the menstrual cycle. The central nervous system plays a role in controlling hormone secretion. Likewise, the target organs themselves, through the process of negative feedback, controls the secretion of hormones. The brain constantly monitors conditions both inside and outside the body. In turn, it triggers the release of hormones as needed. For example, when it is too cold, it triggers the release of thyroid stimulating hormone to promote body heat. After a heavy meal, it stimulates the secretion of growth hormone to break down fats for energy and use amino acids to build tissue. When the pituitary stimulates another endocrine gland to secrete its hormone, that hormone is then fed back to the pituitary, telling it to stop for the release of the trophic hormones. This process is called negative feedback. Negative feedback loops are used extensively to regulate hormones in the hypothalamic pituitary axis. Extreme cold stimulates the hypothalamus to release thyrotropin releasing hormone. Thyrotropin releasing hormone stimulates the anterior pituitary to release thyroid stimulating hormone. Thyroid stimulating hormone stimulates the thyroid to release thyroid hormones. Thyroid hormones stimulate metabolism, increasing warmth. At the same time, the increase the thyroid hormone inhibits release of thyroid stimulating hormone by the pituitary gland. Now we will see the pineal gland. The pineal gland is a small pine cone-shaped mass of glandular tissue found just posterior to the thalamus of the brain. The pineal gland produces the hormone melatonin that helps to regulate the human sleep-wake cycle known as the circadian rhythm. The activity of the pineal gland is inhibited by stimulation from the photoreceptors of the retina. This light sensitivity causes melatonin to be produced only in low light or darkness. Increased melatonin production causes humans to feel drowsy at night time when the pineal gland is active. Melatonin functions as a biological clock and can help to regulate the circadian rhythm, which are associated with environmental day and night cycles and help the body to distinguish day from night. Melatonin is not fully understood but appears to inhibit gonadotropin secretion to help regulate the female reproductive cycle and to control the onset of puberty. In children, melatonin may inhibit early sexual maturation. Large amount of melatonin receptors react to bright light can alter the biological clock. Therefore, altered melatonin levels may result in variations of appetite, body temperature, and sleep habits. Next, we will cover thyroid gland. The thyroid gland is located just below the larynx on either side and in front of the trachea. The thyroid gland consists of two large lobes that are connected by a broad structure called isthmus, forming a butterfly shape, and is covered by a capsule of connective tissue with secretory parts called follicles. Thyroid tissue is made of tiny sacs called thyroid follicles. Each follicle is filled with a thick fluid called thyroid colloid. The thyroid gland produces three major hormones. First one is T3, that is triiodothyronine. Second one is T4, that is thyroxine, or also known as tetraiodothyronine, and calcitonin. The cells lining the sac secrete the two main T3 and T4 hormones. Unlike other glands, the thyroid gland can store the hormones for later use. Cells between the thyroid follicles called parafollicular cells, also known as C cells, secrete another hormone called calcitonin. These cells lie in the follicular epithelium, protruding into the connective tissue surrounding the thyroid follicles. It is secreted in response to rising blood calcium levels. Calcitonin triggers the deposition of calcium in bone, thus promoting bone formation. Calcitonin is a polypeptide hormone that regulates concentrations of blood calcium and phosphate ions, and its release is controlled by blood concentration of calcium ions. Calcitonin inhibits osteoclast activity, which inhibits bone reabsorption and the release of calcium from the bone matrix. It also stimulates calcium uptake and incorporation into the bone matrix. Calcitonin is released when calcium ion levels in the blood rise above a certain set point. So, the function of calcitonin is to reduce the concentration of calcium ions in the blood by aiding the absorption of calcium into the matrix of bones. The hormones T3 and T4 work together to regulate the body's metabolic rate. Increased levels of T3 and T4 lead to increased cellular activity and energy usage in the body. The thyroid hormone is produced by the follicles and stored in colloid. Follicular cells produce glycoprotein called thyroglobulin. From this substance, thyroid hormone is synthesized, which affects nearly every cell in the body. Thyroxine regulates metabolism of carbohydrates, lipids, and proteins, determining the body's basal metabolic rate. Thyroid hormones are required for growth, development, and maturation of the nervous system. Thyroid hormone secretion is controlled by the hypothalamus and pituitary gland. Thyroid releasing hormone from hypothalamus stimulate anterior pituitary gland to produce thyroid stimulating hormone. These thyroid stimulating hormones stimulate the thyroid to secrete T3 and T4. Only 10 to 20% of T3 is produced from thyroid gland. Major portion of T3, however, is produced in peripheral organs like liver, adipose tissue, glia, and skeletal muscles by deiodination from circulating T4. The deiodination is controlled by numerous hormones and nervous signals including TSH, vasopressin, and catecholamines. Both T4 and T3 easily cross the cell membrane and interact with the receptors inside the cell. In the target cells, thyroid hormones stimulate enzymes that are involved with the glucose oxidation. This is known as the calorigenic effect, and its overall effects are an increase in the basal metabolic rate, an increase in oxygen consumption by the cell, and an increase in the production of body heat. Basal metabolic rate is the amount of energy expended while at rest in a temperate environment. That means it is not too cold or hot. The release of energy in this state is enough for the functioning of the vital signs. As basal metabolic rate is increased, so oxygen consumption is increased, as oxygen is required in the production of energy. Iodine is essential for the production of thyroid hormones. Iodine travels in the blood as iodide, which is taken up into the follicular cells by a sodium iodide symbiote. Iodide then travels from within the cell into the lumen through the action of pendrin, which is an iodide chloride antiporter. In the follicular lumen, the iodide is then oxidized to iodine. This makes it more reactive, and the iodine is attached to the active thyrosin units in thyroglobulin by the enzyme thyroid peroxidase. When the follicular cells are stimulated by thyroid stimulating hormone, the follicular cells reabsorb thyroglobulin from the follicular lumen. The iodinated tyrosins are cleaved, forming the thyroid hormones T3 and T4. These T3 and T4 are released into the blood. The hormones secreted from the gland are about 80 to 90% of T4 and about 10 to 20% of T3. Next, we will cover parathyroid glands. Parathyroid glands are 4 small masses of glandular tissue about the size of a grain of rice embedded in the posterior surface of the thyroid gland, with 2 on each side of the trachea. They are covered in thin connective tissue capsules and appear yellowish brown. These glands secrete parathyroid gland in response to low blood levels of calcium. Parathyroid hormone is also known as parathormone. It increases blood calcium concentration and decreases blood phosphate ion concentration to affect the bones, intestines, and kidneys. Parathyroid hormone acts with vitamin D to regulate calcium and phosphorus levels in the blood and bone. Parathyroid hormone is the single most important hormone for the control of the calcium balance in the blood. The major target cells are in the bones and the kidneys. The functions of parathyroid hormones are: it increases intestinal calcium absorption, it stimulates renal calcium absorption, and it stimulates osteoclast activity and therefore reabsorption of calcium from the bones. Normal nerve and muscle function, blood clotting, cell membrane permeability, and the function of certain enzymes all depend on adequate levels of calcium. Physiologically, calcium is important in the transmission of nerve impulses, and it is involved in muscle contraction and is also required in the creation of clotting factors in the blood. The regulation of parathyroid hormone synthesis and secretion is in response to the levels of calcium in the blood, which is monitored by cells in the gland. A reduced blood calcium level leads to an increase in the synthesis and secretion of parathyroid hormone. In an effort to achieve calcium homeostasis, parathyroid hormone exerts its influence on the bones, kidneys, and intestines. Parathyroid hormone inhibits new bone formation while stimulating the breakdown of old bone, causing calcium to move out of bone and into the blood. Parathyroid hormone encourages the kidneys to reabsorb calcium, blocking its excretion into the urine while promoting the secretion of phosphate. Parathyroid hormone also prompts the kidneys to activate vitamin D, necessary for intestinal absorption of calcium. After its activation by the kidneys, vitamin D allows the intestines to absorb calcium from food. The calcium is transported through intestinal cells and into the blood. Calcitriol is a hormone released by the kidneys in response to a decrease in calcium ions in the blood. It is known to have some effect on parathyroid hormone secretion and inhibits the release of calcitonin. It also promotes the absorption of calcium from the gut and the reabsorption of calcium from the renal tubules. Next is thymus gland. The thymus gland is a soft, triangular-shaped organ found in the chest posterior to the sternum. The thymus gland produces hormones called thymosins that help to train and develop T lymphocytes during fetal development and childhood. The T lymphocytes produced in the thymus go on to protect the body from pathogens throughout a person's entire life. The thymus becomes inactive during puberty and is slowly replaced by adipose tissue throughout the person's life. Now we will cover pancreas or the islets of Langerhans or pancreatic islets. The pancreas is a large gland located in the abdominal cavity just inferior and posterior to the stomach. The pancreas is considered to be heterocrine gland as it contains both endocrine and exocrine tissue. An exocrine gland secreting digestive juice and an endocrine gland releasing hormones. Pancreas is an elongated, slightly flattened organ behind the parietal peritoneum. It is joined to the duodenum of the small intestine, transporting digestive juice into the intestine. The endocrine cells of the pancreas is only about 1% of the total mass of pancreas and are found in small groups throughout the pancreas called islets of Langerhans. The pancreas contains millions of microscopic structures called these islets of Langerhans, which are scattered within the gland tissue that produces pancreatic juice. Within these islets are three types of cells that secrete hormones. They are alpha, beta, and delta cells. Now we will see each cell in detail. Alpha cells. Alpha cells secrete glucagon, which raises the level of glucose in the blood, thus protecting the body from hypoglycemia. This is done by stimulating the liver and skeletal muscle to break down glycogen to release glucose into the bloodstream. Between meals, when blood glucose levels fall, glucagon stimulates liver cells to break down the polysaccharide glycogen into glucose and also to convert fatty acids and amino acids into glucose. This process is called gluconeogenesis. The resulting glucose is released into the bloodstream, causing blood glucose levels to rise. Beta cells secrete insulin that lowers blood glucose levels by stimulating cells and tissues to remove glucose from the blood when it is abundant by converting it into glycogen. After eating, the levels of glucose and amino acids in the blood rise. Insulin stimulates cells to absorb both of these nutrients, causing blood glucose levels to fall. Delta cells make somatostatin, which is a hormone secreted in the pancreas and also in pituitary gland that works within the pancreas to regulate the other endocrine cells and which inhibits the release of gastric juice. Specifically, it inhibits the release of both glucagon and insulin and slows the food absorption and the enzyme secretion in the digestive tract. It also inhibits the release of growth hormone. In short, when blood glucose levels are low, alpha cells secrete glucagon, which increase blood glucose level. When blood glucose levels are high, beta cells secrete insulin to decrease glucose in blood. Delta cells in the islets also secrete somatostatin, which decreases the release of both insulin and glucagon. F cells produce pancreatic polypeptide, a hormone that inhibits gallbladder contractions while regulating pancreatic enzyme production. These F cells are also the cells in the pancreatic islets of the pancreas. Glucagon secretion is regulated by negative feedback and it prevents hyperglycemia from occurring when glucose concentration is relatively low. Glucagon is so powerful that just one molecule can trigger the release of 100 million glucose molecules into the bloodstream. Humoral stimuli causes the alpha cells to secrete glucagon, although stimulation from the sympathetic nervous system and rising amino acid levels also play a role. The release of glucagon is suppressed by insulin, somatostatin, and rising blood glucose levels. Insulin decreases blood glucose concentration, promotes amino acid transport into cells, increases protein synthesis, and stimulates adipose cells to make and store fat. Insulin secretion is also controlled by negative feedback, and insulin prevents high blood glucose concentration by promoting glycogen formation. Insulin secretion decreases as glucose concentrations fall. Just after we eat, insulin lowers blood glucose levels and also influences protein and fat metabolism. So what happens after we eat? Blood glucose levels rise as glucose flows from the digestive tract into the bloodstream. High blood glucose levels stimulate the beta cells of the pancreas to secrete insulin. Insulin triggers two reactions. It stimulates the cells to take up more glucose. It causes the liver to take up glucose and store it as glycogen. The combined result is that glucose levels return to normal levels. When blood glucose levels drop below a certain point, such as after skipping a meal, the alpha cells of the pancreas release glucagon into the blood. Glucagon stimulates the liver to break down stored glycogen into glucose, which it then releases into the bloodstream. This causes blood glucose levels to rise. What are the functions of insulin? Insulin enhances membrane transport of glucose into primary fat and muscle cells, inhibits glycogen breakdown into glucose, and inhibits conversion of fats or amino acid to glucose. Because the brain, kidneys, and liver have easy access to blood glucose, no matter what the insulin level currently is, insulin is not required for glucose entry into these organs. In the brain, the insulin plays roles in feeding behaviors, learning, memory, and neuronal development. Insulin and glucagon function together to maintain stable blood glucose concentration, even though the amount of carbohydrates taken by a person may vary widely. Nerve cells are partially sensitive to blood glucose concentration changes. Such changes can alter brain functions. Prior to the development of type 2 diabetes, most patients develop prediabetes. That means blood glucose levels that are higher than normal but not high enough to be diagnosed as diabetes. Prediabetes may be referred to as impaired glucose tolerance or impaired fasting glucose. This condition increases the risk for developing type 2 diabetes and cardiovascular disease.